You have two ages. One is your chronological age, the candles on your birthday cake. The other is your biological age, how old your body actually is at the cellular level, which can be years older or younger. The tools that measure this, called epigenetic clocks, are one of the most important and hyped developments in longevity. Let's understand how they work, and how much to trust them. To understand these clocks, you first need one idea: epigenetics. Your DNA is the hardware, the fixed sequence of letters you're born with, which barely changes across your whole life. But sitting on top of that DNA is a layer of chemical marks that act like software, switching individual genes on and off in each cell. The most studied of these are called methylation marks, small chemical tags attached to the DNA. Here's the crucial discovery: as you age, the pattern of these methylation marks changes across your genome in a remarkably predictable, clock-like way. Certain spots gain marks, others lose them, and they do so on a consistent schedule. That predictability is everything, because if a pattern changes on a reliable schedule, you can read it backward to estimate age. So how does the clock actually work? Scientists took DNA methylation data from thousands of people of known ages and used machine learning to find the specific set of marks that, together, best predict a person's age. That trained model is the epigenetic clock. Now you can take a sample, usually blood or saliva, read its methylation pattern, feed it into the clock, and out comes a number: your biological age. And here's what makes it powerful and a little unsettling. If your biological age comes out higher than your actual birthday age, it suggests your body is aging faster than average. If it comes out lower, you're aging more slowly than the calendar says. It's an attempt to measure not how long you've lived, but how worn your biology actually is. For a field that struggled for decades to measure aging at all, that's a genuine breakthrough. There isn't just one clock, there are several, and they measure subtly different things. The original, from 2013, is often called Horvath's clock, after the scientist who built it, and it's foundational. Then came more refined versions. GrimAge is trained not on age but on time-to-death data, which makes it currently the strongest single predictor of mortality risk. PhenoAge blends methylation marks with standard clinical blood markers for a more health-focused read. And a particularly clever one, Dunedin Pace, doesn't give you a static age at all, it estimates your current rate of aging, measured in biological years per calendar year. A healthy person might score around 9/10, meaning they age slower than the calendar. An unhealthy person might score 1.2, aging faster. Because it measures your pace right now, Dunedin Pace is the most responsive to whether an intervention is actually working. Now the honest caveats, because this is where hype runs wild. First, precision. These clocks are superb for studying populations and research, but for an individual consumer test, they can be surprisingly noisy. The very same DNA sample, tested twice, can give meaningfully different numbers, and results vary between labs. So a single reading is a rough estimate, not a precise verdict on your body. Second, and more important, the clocks show correlation, not proven causation. We know that a higher biological age is associated with worse health outcomes. But it has not yet been proven that if you take an intervention and your clock number goes down, you'll actually live longer or healthier as a result. The clock might be a readout of aging without being the thing that, when changed, changes your fate. So use these as an interesting trend to track over time, not a scoreboard to obsess over, and be deeply skeptical of anything promising to reverse your epigenetic age. This is educational, not medical advice.